So, now that we have reviewed what happens here at our postsynaptic cleft with our axon terminal and our body cell, let's discuss its implications in pharmacology, and let's go through the rest of chapter 12, and hopefully it'll make a little more sense. So, I'm going to pull up our slideshow here. Our book says that the transmission of a neurotransmitter starts in this cell, and not just with the action potential, that it starts when this cell makes the neurotransmitter. So we talked about the fact that if it's in these vesicles, it's not really doing anything, but the thing that we need to understand is in order for it to do something, we have to have it stored here in this vesicle. We can't need to make it when we need it. We have to have it stored here and floating around so that when our body needs it, it's ready to roll. That's an important thing. So our book views that, it being there, ready to go in the vesicle, as the first stage of transmission, or the synthesis of the neurotransmitter as the first stage of transmission. The second is the storage, so getting it into these vesicles and having it ready for use. It views the third phase of transmission as the release when it pops out of its little vesicle. The fourth as receptor binding, when it binds to our little spot on the cell wall. And the fifth as the termination of transmission, so when it stops. If you remember, when we studied the neuromuscular junction, part of that transmission from the axon terminal to the cell wall and it being effective is that acetylcholine is not left here in the postsynaptic cleft, that it is terminated here. So some cells, I believe, actually reuptake the neurotransmitter, and that function will vary across different cells in the body. But if we had acetylcholine, for example, in the neuromuscular junction available in the postsynaptic cleft all the time, we would have tetanus, like a continued stimulus, and it wouldn't be effective. It would just be one long muscle stimulus, and we wouldn't have that relaxation. So we have to have the neurotransmitter termination. So we talked about the transmitter stimulus or synthesis. So one way that drugs could alter neurotransmission would be to have more neurotransmitter available. So some drugs, their function is actually to increase the synthesis of neurotransmitter, to make more of that chemical, to have more here in the cell ready for use, to have more available. Some decrease transmitter synthesis. So some of them are going to make less available, maybe just one or two if we want to alter that transmission. Some are going to, we said, cause more synthesis. Some cause receptor activation to decrease. So some are going to cause less activation of our receptors on the cell in the postsynaptic cleft. And some promote or inhibit. So one example of one that would promote would be amphetamines, right? We get all worked up with those. And then botulinum toxin would be an example of one that is inhibitory. The effects of drugs on the synaptic transmission. So remember we said some cause activation. And in our last video we talked about just because it activates a receptor doesn't mean that the physiologic function speeds up. And a great example of that we said was acetylcholine at the heart. So there's a receptor for acetylcholine on cardiac muscle tissue, and that receptor is activated by acetylcholine. But the activation of that receptor by acetylcholine slows the contractility of the heart down. So just because something is activated doesn't mean things are speeding up. It could slow it down or it could speed it up. It could happen either way, but that means that that receptor is activated. So, an example of receptor activation, we said, acetylcholine at the heart. Another example is morphine, we said. So, morphine activates those opioid receptors. And we said Narcan comes in and blocks it. The termination of transmission causes receptor activation to increase. and it blocks transmitter reuptake. So I don't know what that means, but it's on our PowerPoint. I think it's just what we were talking about, the antagonist versus the agonist and how those are acting at the receptor. So we said drugs that alter the action potential moving down the axon, those are not selective. They can attack everything in the body, or impact everything in the body that's affected by a neuron with action potential. That's a pretty big group of things. So the ones that we want to alter, the ones that we're going to talk about most in pharmacology, the ones that drugs are going to alter are these neurotransmitters. And the more receptors there are for that particular neurotransmitter, the less selective a drug is. So if I'm making more of acetylcholine everywhere in the body, then everywhere that uses acetylcholine is going to have more of that. And there's a lot of places in the body that use acetylcholine. So that's not very selective. If let's say, if there's a particular neurotransmitter that only affects the heart rate, and we make more of that, then it's going to be very specific. It's going to be very selective. So the thing to take away from that is we want to find the neurotransmitter that affects what we want to alter. The more selective it is, the more we can alter that drug, the better off we are. But we can't always do that, right? Because ideal drugs, they don't really exist. They all have side effects. But the more we understand our neurotransmitters and the more we can pick which one we're altering, the more selective our drug is. So we need to understand the receptors throughout the body. So we know what chapters, that's pretty much everything for chapter 12. I was just reading the rest of Ms. McCarty's notes. So we understand the basic function of the axon terminal and the receptor in the peripheral nervous system. So chapter 13 goes into the types of specific receptors, because remember, the more specific, the more ideal our drug is. And our next video will start on that.